and thank you very much for joining us here today. Welcome to a new edition of the EurJOC Virtual Symposium, and this time a special edition in collaboration with the Organic Division of the French Chemical Society. My name is Anne Nice, and I'm the Editor-in-Chief of EurJOC. And with me today is Charlotte Gerspanta, Deputy of the Journal. It's our pleasure to host this event together with Matthieu Sollogoub, who's the President of the Organic Division of the French Chemical Society. And he's joining us from Paris today, together with a few of his colleagues. We're all very excited about the lineup of speakers that have agreed to present today at this virtual edition of the Journeé DCO. And we look forward to fantastic talks by Thomas Carell, Jean-Francois Nierengarten, Xavier Bugaut, Ludwig-Maximilians-Universität, and Francis Arnold. The event is brought to you by Chemistry Europe, which is EurJOC's society publisher, of which the French Chemical Society is also a member of. Before we start with the scientific program, I would like now to hand over to Matthieu, who will say a few words about the Organic Division of the Society, its prize winners, and also about the symposium. So Matthieu, please start.
Thank you very much, Anne, and it's really a pleasure to have you all with us today, and to this. It's an experience for all of us, and I hope it will be successful, and maybe we can carry on this experience. We're really happy to do it together with EurJOC and Chemistry Europe. So this is what replaces one of our biannual days, Journée de printemps and Journée d'automne, because of the circumstances that you know. So it is not only me, this is the organization of a bureau, but today this bureau has two new members for today, Anne Nice and Charlotte Gerspanta from EurJOC, and we are really grateful for their help and for providing all the means to do this this event. So, as Anne just said, we will have two prestigious conferences from great scientists, Thomas Carell from München and Francis Arnold from California Institute of Technology. And also we are have the pleasure to present to the community the recipients of our prizes. So the biggest prize of the French Chemical Society Organic Division is the prize, the DCO prize, which was awarded to Jean-Francois Nierengarten from University of Strasbourg, and he will talk just after Thomas Carell. Then we have the Prix Jean Normand to Xavier Bugaut from Marseille University, and finally the Prix Marc Julia to Ludwig-Maximilians-Universität from Rennes University. So you actually can see that we have, if you know French geography, it's the three corners of France that receive almost prizes this year. So it's just a reminder that the Organic Chemistry Division of the French Chemical Society is here to promote you. There will be some prizes. Every year we are giving some prizes. Actually, we are giving 8 prizes. Half of them is presenting today. Half of them as presented last year. Please apply to those prizes. You just have to be member of the French Chemical Society. So, thank you for your attention. Now we are going to the scientific part of this, and I would like to thank our two first speakers, Thomas Carell and Jean-Francois Nierengarten, to participate even in such time for them and for us because of the early passing of François Diedrich. And so I hand you back the Anne, you can take over now.
Thank you very much, Matthieu. And before we start with the scientific program and here with the first talk by Thomas Carell, just a few general remarks from my side. So I have the pleasure of introducing all the speakers today, and Matthieu will afterwards moderate the question and answer session. At the middle of the event, we're trying out a new section called Meet the Chemists. And here you have half an hour to pose your questions to our first two speakers, and that is Thomas Carell and Jean-Francois Nierengarten. So please get your questions ready. And to do so, please use the chat function, which you can use at the bottom, or which you can see at the bottom of your screen. So here you can also chat amongst each other, but if you want to ask a question, please tag it with @Thomas and Jean-Francois to help us identify them. Please also engage with us on Twitter. We have a very good Twitter followership and like to engage with you there. We're always happy to see how you are watching the event and to know if you're actually enjoying the event. So now I don't want to keep you waiting for too much longer, and I would like to introduce the first speaker, which is Thomas Carell, and I would like to ask him to turn on his camera and come on screen. Thank you, Thomas. So Thomas started his academic career in chemistry at the University in Münster, and then he continued in Heidelberg, where he obtained his PhD in 1993 with Professor Stab. After a postdoctoral research stay with Julius Rebeck at the MIT, he moved to the ETH in Zurich, where he started his independent research in the group of François Diederich. After completing his habilitation in 1999, he took on a full professorship of organic chemistry in Marburg, and in 2004, he moved to his current position at the LMU in Munich. Thomas is the recipient of many awards, such as the Otto Bayer or the Leibniz Prize, and he's also the chair of the editorial board of our sister journal, ChemBioChem. His work focuses on studying various aspects of epigenetics, and today he will tell us more about the second dimension of the genetic code. So Thomas, we look forward to your talk, and I would like to ask you now to share your screen.
Now I'm trying to share my screen.
Yeah.
But I cannot. Teilen? No.
So if you do, just worked in the rehearsal. Can you share your application?
Let me see again.
Yeah. No problem. So if you click the blue, if you go into presentation mode now.
Yeah, I'm just starting it once more.
Mm-hmm.
Share your screen. No?
No, we just tested it 10 minutes ago. Yeah.
That's right, but it doesn't. Can you see it now?
Yes, now we have it. Perfect.
All
Thank
right.
you very
So
much.
here
No problem.
Also, it's very short. Yeah, so first of all, Anne, thank you very much for the kind words of introduction. I'm also thanking Matthieu, Chemistry Europe, and the wonderful journal EurJOC for enabling this conference. Of course, a Zoom conference cannot, or whatever, these internet conferences cannot replace personal meetings. On the other side, they have the wonderful advantage that even people can join from far remote places, so there are advantages and disadvantages. I would like to dedicate this lecture to François Diederich, one of the most eminent organic chemists that we had, and Jean-Francois and me, we both overlapped in his group, so I'm very thankful to François because he taught me so much about organic chemistry, and I think I wouldn't be what I am right now without having been in his lab. So let me dedicate my lecture to François. I'm going to talk a little bit about the second code in DNA, and before I'm starting to do science, let me thank a few people. I would like to thank Fabio. He is a stem cell biologist in the group, and also Marcus as a biologist. They are both helping me to do good biology. I like to thank a lot of co-workers, synthetic chemists like Toni Pfaffeneder, Angie Kirchner, Mirko Wagner, Katharina Iwan. They have synthesized many molecules. They did a lot of mass spectrometry. I'm going to talk about the results in a bit. René Raimhoff is still postdocing in the US, another wonderful chemist. Arne Schröder, Barbara Steigenberger, a mass spectrometrist. Alexander Schön has made a lot of molecules. Evelina has made many molecules, and also this Evelina from Finland has contributed to the research results I'm presenting. And let me already now thank my university, LMU Munich, for providing an outstanding research environment for all of us. So what do I mean with the second genetic code? First, we all know that the genetic information of most organisms is stored in the form of this DNA double strand. And the DNA double strand has two important features. First, it provides a sequence of these 4 bases. I mean, a computer is using a sequence of 0 and 1 to encode information. Our information density is a lot higher instead of 0 and 1, so that's a 2-letter code. We are using 4 letters in order to encode information A, G, C, and T. So the sequence of these 4 bases is basically the information that is called the genetic information. The second feature of the DNA double strand is that this sequence code is self-complementary, meaning that the letter A can base pair selectively with T and vice versa. And the same is true for G and C. So we have a sequence of these 4 letters that encodes information, and the information can be copied and therefore transferred to the next generation because of the self-complementarity or the complementarity of the genetic system. This is what we learn if we look into biochemistry textbooks. And what we forget is, since years, we know there is a 5th base of the genetic system. This is 5-methylcytosine. And this is already an addition, and it's somehow the starting point for a second layer of information. In 2009, another base was discovered, actually a relatively large extent, 5-hydroxymethylcytosine. So all of a sudden, the 4-letter code that we know from textbooks, from biochemistry textbooks, was extended to a 6-letter code. And what we did already in 2010, this is the 3rd paper in the whole field, is we wanted to know how widely distributed is this 6-base hydroxymethylcytosine. Is it present only in certain parts of tissues, or how widely is it distributed? So what we did is we took a mouse brain. This is a mouse brain. We dissected it into the various parts: cerebellum, brainstem, hypothalamus. And then we were using mass spectrometry technologies that I will explain in a bit in order to quantify the levels of hydroxymethylcytosine and 5-methylcytosine. And this is more difficult than one thinks, because mass spectrometry is per se not a quantitative method. So we had to synthesize isotope standards to achieve this quantification. And what we see is that in all these tissues, the 5-methylcytosine content is relatively stable at between 4 and 5%. But we also see that hydroxymethylcytosine is basically present everywhere in the brain. And the levels are relatively high, with 0.7% of all the cytosines being in this hydroxymethylated version. And the situation becomes even more paradox if when we started to analyze mouse organs. Here we see again methylcytidine, the 5th base of the genetic system, is present at between 4 and 5%, relatively stable in all different tissues, while hydroxymethylcytosine is also present everywhere. This is the first surprise. The second surprise is again relatively large amounts in brain. And the third surprise is that in contrast to methylcytosine, we get a very uneven distribution. We have tissues with low, medium, and brain tissue with very high levels of hydroxymethylcytosine. So very interesting. The base is really everywhere. And since 2010, we can call hydroxymethylcytosine indeed the 6th base of the genetic system. Of course, 2010, 2011, everybody was asking, is this all, or are there other bases that have been overlooked so far? And indeed, in 2011, these 3 groups independently from each other discovered 5-formylcytidine and 5-carboxylcytosine as additional bases of the genetic system. So all of a sudden, we have now a code 4 plus 4. We have A, G, C, and T, which establishes the sequence information. And then we have Regarding the C base, we have methyl C, hydroxymethyl C, formylcytidine, and carboxyl C. So we have 5 additional bases, and the discovery of these new bases was again done by mass spectrometry based on the high resolution mass spec of the compound that was isolated from DNA, and synthetic material was used as an internal standard in order to establish the correct retention times and to get the fragmentation pattern. So there is no doubt that these 2 bases exist as well. These are human data, so we are now jumping into the year 2015. We see again hydroxymethylcytosine has not only an uneven distribution regarding the various tissues, but it is also increasing with lifetime, with age. So a 15-week-old unborn human, these are human data, has a very low level of hydroxymethylcytosine. Then with age, this increasing. We see again, methylcytosine is much more stable, so not only stable in different tissues, but also stable over age of the organism. And here we see formylcytosine is also nicely present in our brain. So our brain tissues are particularly rich in hydroxymethylcytosine. Not only brain tissue, but also very early on in life. So here we are jumping into the process of fertilization when a sperm cell is fertilizing an egg cell. So this is how life is starting. We all have started from this event. And of course, now in this zygote, we are merging the parental DNA and the maternal DNA to form a new organism. And if we now use antibodies, a red one against methylcytosine and a green one against hydroxymethylcytosine, and we zoom into one of the zygotes, what we see is that the maternal DNA still contains methylcytosine, while the paternal DNA is now moving from methyl to carboxylcytidine. So this picture contains a lot of information. It tells us that hydroxymethylcytosine is generated from methylcytidine, so it's really an oxidation event that creates the 6th base of the genetic system from the 5th base of the genetic system. And in the meantime, also the enzymes have been discovered that do this in our genetic material. These are called 10-11 translocation enzymes. These are alpha-ketoglutarate-dependent oxidases. And in addition, what we learn is that our life starts with this interesting chemistry that is separated between the maternal genome and the paternal genome. So this is the genome from the father, which is selectively performing this oxidation reaction, basically eliminating methylcytosine content and converting it to hydroxymethylcytidine. And this is continuing even if now the new genetic system is forming. We see now formation of the chromosomes. Here the mother DNA is still red, full of methylcytidine, but it's also turning a little bit green here, so the oxidation is now also going on. So what do we see here at the beginning of life? We see here that these 2 genetic systems, from the mother and from the father, from 2 different organisms, they merge and they need to orchestrate the functioning of these genetic system in order to create a new organisms. Now, in the meantime, in the last couple of years, we did a lot of research. We saw that this oxidation chemistry is very important for the degeneration of tissues to cancer tissues. We found, using this proteomics approach, we found specific proteins binding to hydroxy, formal, and carboxylcytidine. So there are proteins that bind to these bases. They read out this additional information and translate it into a biological function. We have now phosphoramidites of all these building blocks that allow us to make DNA containing these new bases in order to study biophysical properties. Today, I would like to focus at one specific story that has kept us busy over the last years. The question is really, what is the function of these bases? And what we currently believe is that these bases encode the activity of a specific gene. I mean, genes are the genetic system is divided into genes. These genes need to be read, they need to be translated into proteins. And of course, a neuron needs other proteins than a fibroblast. So we have 250 different cell types in our body, skin cells, gland cells, muscle cells, neurons, and they all need to make different proteins. So they have to have different activity states of the different genes. Genes that in the fibroblast may be on have to be switched off or dimmed down in the neuronal cell. And in the neuron, we need specific neuronal proteins that need to be manufactured, while proteins that are important for a fibroblast are not required at all. So we need to regulate the transcriptional activity of genes. And we believe that this activity regulation is established by these new bases with cytidine switching a gene on, methylation of cytidines in control elements of genes switches the gene typically off, and then further oxidation, we know in the meantime, to hydroxymethylcytosine puts the activity into a super on state. So now the gene is really massively transcribed. And then we believe that formylcytidine and carboxylcytosine are more transient bases that allow the whole system to move back. So we have on, off, super on, and then with these bases, we somehow move back into the original state to basically start again. And this allows cells during differentiation to switch genes in an active or into a passive mode. And the question was really, what is the chemistry that occurs here from these bases back to cytidine in stem cells? So this is what we proposed. We proposed that there is this oxidation, first methylation, and then the oxidation cascade. And then the idea is that formal and carboxylcytosine are both bases that upon reaction with a nucleophile, and there are many enzymes that work on DNA that have an active nucleophile, a thiol, we propose that if a thiol is reacting here with this Michael system, that we get up subsequently a decarboxylation reaction, or after hybridization of the formal bond, something like a deformal reaction, which allows these two bases to move back to C. So we have C, methyl C, so on, off, super on, further oxidation, and then reaction by a nucleophile, followed by deformylation or decarboxylation gives us back the cytidine. So we published that this is indeed possible. If you treat these bases, we used synthetic material with nucleophiles like thiols. Indeed, these bases deformulate and decarboxylate, but the question was, does that really take place inside stem cells? And in order to investigate this question, you need a microscope that allows you to discover, uncover, unravel chemistry in the nucleus of a developing stem cell. And what we do, what is our camera? What is our microscope? Our microscope is again mass spec. So what we do is we take these stem cells and we feed them with isotopically labeled material, where we exchange the C12 by C13. And then we have here maybe formal or carboxyl. And if we are lucky, and we need luck here, if we are lucky, then this synthetic material, if we add it to the culture medium, is taken up by the cells and is randomly incorporated into the genome. And then we have probe molecules sitting in the genome. What we then can do after certain time intervals or during differentiation, we can isolate the DNA from the material, and then we get, of course, then we can digest the DNA material. Then we get the individual bases. These are the bases that establish the sequence code. Then we have, of course, MC, HMC, FC, and CSC. So this is the epigenetic code. And, of course, we also get our incorporated base that we added here. And because the base has a different distribution of C13 and N15 atoms, we can distinguish it by mass spec from all the other bases. And, of course, we can then look for new bases that have been formed from this parent base. And this allows us then to study the chemistry, for example, the chemistry that goes from X to Y at random positions in the genome. Let's go into the experiment. So what we were synthesizing is this double N15, 5 times C13 labeled formal cytosine. The idea is feed it to stem cells, hope that it's getting incorporated into the DNA. And if deformylation occurs, so if this bond is cleaved, then we should see it after isolation of the DNA, digestion, and analysis, because we would get a new cytidine derivative that is different from the canonical cytidine by 2 N15 and 5 C13 labels. So if we would be able to detect a cytidine that is 7 units heavier than the natural cytidine, then we know this CC bond cleavage has occurred. To this end, you need to synthesize the compound, and I don't have time to go into the synthesis. I just tell you that synthetic organic chemistry is absolutely essential to enable these experiments. We start here with fully labeled glycose, which we convert into fully C13 labeled ribose, and then we use urea that is double N15 labeled to build up the uridine base. We subsequently perform a Vorbrücken nucleosidation to make the nucleoside. Then comes protection group chemistry, Barton-McCombie deoxygenation. We are converting to the U into the C base. We use now metallogenic chemistry, first iodination, metallogenic chemistry to establish the formal group, and subsequently we cleave the protecting group. And here we are, after many steps of synthesis, we are getting to this specifically labeled compound 1. And indeed, this compound was then fed. We added to our stem cell cultures. And we were really lucky that stem cells, because they divide so quickly, they take up whatever they get. And they also took up randomly this formulated double N15 5 times C13 labeled cytidine derivative. When we isolated the DNA and we looked for this base, we could clearly detect it by mass spec. So it's incorporated into the genome. And now the question was, do we see also the cytidine compound? The only possibility to get a cytidine compound with a +7 mass signature is by deformulation. And this is indeed the case when we use just normal stem cells without any feeding. In this HPLC chromatogram here, the black curve, we see absolutely no signal. If we, however, feed the formal cytidine derivative, we see at 1.95 minutes a new signature coming up. And this new signal comes from the deformulated cytidine derivative 2. And then we even synthesized a more heavily isolated compound that we co-injected. And this C compound eludes at the same time. And based on the fragmentation patterns, we can then say that the red compound is really this one after feeding, and this gray curve here contains this species, which is also fragmenting. So we know no feeding, no signal. Feeding of formulated cytidine derivative, we get a wonderful new signal, and this is because of deformulation. So this is again summarizing the data. We get incorporation of the formulated labeled cytidine derivative. It's nicely deformulating. We see also a couple of other processes that occur which are still under investigation. In order to be absolutely sure that this deformulation chemistry takes place, we also changed a little bit the strategy by synthesizing a compound that is double N14 and 15 labeled, which contains in addition a fluorinated group. The reason is that we saw a lot of DNA repair going on that removed our incorporated base. And in order to block these repair processes to get higher concentrations of these bases in the genetic material, we incorporated this fluorine atom. The fluorine atom is basically blocking repair processes that eliminate our probe molecule inside the genetic material. Again, we needed to synthesize the compound, and here it is. We synthesized also the formulated, but the formulated fluorinated double N15 labeled cytidine derivative. Here's the final compound. We also made the hydroxymethyl compound, but for this study, the formal compound was needed. We feed it again to stem cells. We hope that it's getting incorporated. And the idea was, is it deformulated? If yes, we should be able to detect a fluorinated cytidine derivative. The only way to make a fluorinated cytidine derivative in the genetic material is if this is incorporated by some kind of deformulation. And indeed, these are the data. If we take the DNA from the stem cells, we digest it into the nucleosides, and we analyze them by mass spec, we see the compound 15 is incorporated. This is clear because we were feeding it. We also see the compound 10. This is this compound. So we see clearly again deformulation. And we also see now, and this is the beauty, we see now remethylation. So we see this cycle occurring, formal cytosine going back to carboxyl cytosine, and then we see even remethylation of these positions, so switching the gene into the off state again. Very recently, we also looked at decarboxylation. Here we needed a slightly different strategy. We synthesized the triphosphates, carboxyl cytidine, again, a fluorinated species. We used a transporter molecule that was reported by Kraus in Angewandte Chemie 2018 in order to move this compound into the stem cell. And then indeed the compound gets nicely incorporated into the genetic material. And the question was then again, do we see now decarboxylation? And the answer is yes, we see it. Here is the signal, very fresh out of the press. The result is 3 weeks old. We see nicely the decarboxylated fluorinated cytidine derivative. So let me summarize a little bit the data. What have we seen? What have we discovered? Well, we have discovered over the last 10 years that we have, of course, sequence information, A, C, G, and T. But we also have epigenetic information. The cytidine can be methylated, hydroxymethylated, formulated, or carboxylated. We have learned that the presence of the various bases determines the activity of the gene, or whether the gene is silenced. For example, fibroblast-specific genes in a neuron. These genes are silenced, so they contain a lot of methylcytosine. And we have learned how this silencing can be overcome by oxidation. This now means full activity of the transcriptional process, further oxidation, and now we have seen deformylation and decarboxylation chemistry moves the whole system back into formal cytosine. And this is, by the way, particularly important for learning. We know that learning goes in hand with strengthening of neuron-neuron connections, increasing the amount of synapses. So this is part of the learning process. While in neurons, most genes are in a passive, in a deactivated states, neurons don't divide anymore. In order to achieve this, certain genes need to be activated. And we know now this for learning, it's very important to reactivate silenced genes. And because these cells don't divide, the only way to reactivate silenced genes in neurons is by oxidation, deformylation, and decarboxylation, or by some kind of a repair process. With this, I'm at the end. I would like to thank my collaborators, particularly Stylianos Michalakis from pharmacy. He provides mouse tissue. I'd like to thank Dr. Kretschmer for providing the human tissue, the analysis I showed at the beginning. The proteomic studies were done with Professor Vermeulen in Nijmegen. I need a lot of theoretical chemistry in order to understand the chemistry that is underlying deformylation and decarboxylation. I do this with my colleagues Christian Ochsenfeld and Henrik Zipse. And I'd like to thank these organizations for providing money for research. And last but not least, my whole research group for enthusiastic research. Thank you very much. Thank you for your attention. And thank you, the organizers, for putting this wonderful conference together.
Thank you so much, Thomas. We can have an applaud here because we're a few people. So there have been quite a few questions on the chat, so I will try to convey them to you. So one question is, why is it always or maybe only C that is modified?
That's a very good question. I think there is no proven answer. What I believe is that it is absolutely critical to, I don't know, do you see me, by the way? Do you see my face? Can you?
You're still sharing your screen, Thomas? Yeah, now we can see.
Now you can see me. Okay.
Thank you.
Okay, here we go. Okay, so I think the most important thing is that we need to be able to transfer the genetic information to the next generation. So we have to make super precise copying. And copying means a C has to be read as a C, and an A and a G and a T correspondingly. So I think nature uses only one position in order to enable accurate copying of the genetic information. If you would place modifications everywhere, then you have to make the polymerase, the copying enzyme, a little bit more sloppy, and then you pay for it with more mutations. So I think the polymerases are designed to be only sloppy on one specific position. That's the C5 positions of C. So there is a little bit of space in the enzyme which allows to encode additional information without jeopardizing the correct copying of the genetic information. So it's a little bit a compromise between having very little variability to make copying extremely precise, and on the other hand, needing a little bit of variability in order to program the genetic information.
Yeah, thanks. I think that also answers another question, which was, can the C be methylated at the other free position? Was it ever looked at? And if it's not possible, why only this position? So you probably just answered this question. And so it's not only based on reactivity, but maybe reactivity plays a role here, no?
Absolutely. The C5 position is easy to activate with a nucleophilic addition to C6. Then you can methylate C5, you eliminate the helper nucleophile, and then you place the methyl group. And this is exactly how the DNMT enzymes, the methylating enzymes, work. It's very difficult to manipulate the C6 position. We did this recently, just as chemists. We wanted to know, do these C6 modified bases exist? They can be made, but you have to build up the whole ring system, so it's relatively complex.
Yeah, we've got a few other questions. So in the order, one question was, if the HMC is increasing with time, almost not present for a child and increasing with age, was it ever established linked with aging or diseases like Parkinson, for example?
Yes. The data are very rude here. So the answer is yes. This epigenetic information is particularly important. We know it's wrong. It's off in diseases like depression and schizophrenia. So these were diseases in the past where we had no biochemical evidence of what's going wrong. Now we know that these seem to be diseases that are associated with changes with wrong methylations and wrong hydroxymethylations. Yeah, and it's increasing with age. I believe that also if you compare mouse with human, the presence of hydroxymethylcytidine had a lot to do with higher cognitive functions, and of course these higher cognitive functions develop with age. So this is why I think it's not a surprise that in humans we reach a maximum level with 22, 23, when our brain has reached its maximum mental capacity. And then it's basically flat and constant.
Thanks very much. So there are many questions on the chat, so I guess I won't have time to ask you them all. So maybe a last question, and again, it's in the order of appearance. Do we know exactly what purpose CAC and FC serve? You spoke of transient bases, but I'm still a little unclear for.
Yes, and this unclearness is well founded. We are also puzzled. We know there are specific proteins that bind to FC and CAC. So we believe that these bases are not only transient bases that are formed during a deformylation or decarboxylation. We know they are relatively stable. And at least for formal cytosine, it has been shown that it forms a covalent bond with the lysines of the histone proteins, and that it is really important in order to pack the DNA in around nucleosomes. So it's really important in order to, for packaging and for organizing the genetic material in the nucleus. It's not that the material is sitting just in the nucleus. We know the genetic material is highly organized in the nucleus. We don't know how this happens. We don't know how order is established, but formal cytosine is important to establish order in the nucleus. That's what we know.
Okay, thank you very much, Thomas. I think we're going to stop now. You can answer questions through the chat if you have access to it. That would be nice. I think there are many questions, and thank you for everyone for asking those questions. That's very, very nice to make it very much alive. And thank you again, Thomas, for a great talk. Okay. Crowded audience.
Yeah, thank you very much also, Thomas. And then we look forward to seeing you again later for the Meet the Chemist session. And now I would like to introduce our next speaker of today's event, and that's Jean-Francois Nierengarten. And I can already see him already on screen. Welcome, Jean-Francois. So Jean-Francois studied biochemistry and chemistry at the University in Strasbourg in France, and he received his doctoral degree in 1994 under the supervision of Jean-Pierre Sauvage and Christiane Dietrich-Buchecker. He then worked as a postdoctoral fellow with François Diederich at the ETH Zurich, and in 1996 he obtained a CNRS researcher position. Currently, he is Directeur de Recherche at the CNRS and also head of the Laboratoire de Chimie des Matériaux Moléculaires in Strasbourg. He has won numerous prizes. For example, in 2019, he received the silver medal of the CNRS, and I'm actually very honored that I was part of this award ceremony, which took place in Strasbourg last year. He's also the winner of the Prix DCO, so the prize of the organic division of the French Chemical Society. And as such, he will present his lecture today. I should also mention that he's the chair of the editorial board of Chemistry, a European journal, and also a fellow of Chemistry Europe. His research focuses on the chemistry of fullerenes for applications in material science and biology. And this is also the topic of his talk today. So Jean-Francois, we very much look forward to your 2019 DCO prize winner lecture. Thank you.
Thank you very much. Matthieu, do you want to tell something? No, it's fine. So
I will tell something afterwards.
Okay.
If you could share your screen, yeah. Okay, we can see your presentation.
Yes. It's working well?
Yes.
Okay,
yeah,
perfect. So I can start.
Thank you.
Okay, so thank you very much, Anne. Thank you very much, Matthieu, and thank you very much for the division of organic chemistry of the French Chemical Society. It's a big honor to receive that award and to deliver that lecture today. Although it's a sad week because, as already Thomas mentioned, last week François Diederich passed away. I would like to dedicate this lecture in the memory of François. He was an outstanding scientist. I think the perception of supramolecular chemistry and weak interactions also in medicinal chemistry changed completely because of the work of François. He's also a precursor in many fields like carbon-rich nanomaterial, Fullerene, chirality, and many other things. We will miss him a lot. So I was very fortunate to work in his group as a postdoc during 2.5 years in Zurich. And in his group, it was great because I think the postdoc is the best moment in your career. You don't care about anything else than science. And when you have an enthusiastic boss like François, it's just a pleasure to do science. And of course, when I obtained my CNRS position in Strasbourg, I used the chemistry I learned in Zurich, Fullerene chemistry, to develop new materials in particular. In the beginning, we were mainly focused on the organization of fullerenes, and we incorporated C60 in liquid crystalline material. And for that, we prepared supramolecular complexes with cyclotriveratrilane receptors. You form a kind of Big Mac. You have two receptors that encapsulate the fullerene, and then you have self-organization into a liquid crystalline phase. We also prepared a lot of amphiphilic fullerene compounds that we organize on the air-water interface in longer films, and then we could transfer them eventually in longer blood-jet films. Those compounds were not just amphiphilic compounds. We used them a lot as building blocks to construct big dendrimers, fullerene-rich dendrimers. That's an area in which we worked a lot. Was mainly the work of a very talented postdoc in the group. It was Uwe Hahn, who prepared huge compound with a lot of fullerene molecules, which is a big challenge because you have to solve issues like solubility, and the characterization is also quite difficult. We also used fullerene a lot for its electronic properties. The fullerene is a good electron acceptor, and we combine C60 with all kind of electron donors, conjugated system, porphyrins, transition metal complexes. And in that particular case, it was mainly because we had a very good interaction with the group of Nicola Maroli in Bologna to perform all the photophysical studies that we could really achieve all those projects. I will just show you one example. When we combine C60 with conjugated system, that's very interesting because you can modulate very easily the electronic properties of the conjugated system by changing the length of the system, the substituents, and those compounds found some applications in the field of solar cells. And we have been the first to show that you can use a single molecule as a photovoltaic molecule. If you sandwich such a molecule in between two electrodes, you can produce electricity with light. So how is that working? This is illustrated in that movie. So you sandwich the molecule in between two electrodes, two different metallic electrodes to have a difference of potential. Upon photoexcitation, eventually you have electron transfer. You produce a positive and a negative charge. They are then going to the electrodes. You have charge injection, and in this way you create a photocurrent. The use of molecule is very nice because you can easily correlate the structure and the photovoltaic response, and in that way you can improve the photovoltaic response by a rational molecular design. I will not go further, but we were very active in that field for years, and more recently we started to use the fullerene not for its electronic properties, but simply as a scaffold. In the particular case of fullerene hexaadducts, you see that you have 6 arenes grafted onto the fullerene core, and you have 12 functional groups around it. It's a globular system, very compact, 3-dimensional, and in principle, you can use it to prepare multifunctional material. But the first step is the chemistry. You can produce those hexaadducts in a single synthetic step, but the yield is very sensitive to the nature of the R group of the malonate group. The reaction is working very well when the malonate is simple, but as soon as the malonate is a little bit fancy, the yields are terrible, the purification are terrible. So the chemistry is there, but you cannot really use it to produce material because you are very quickly limited by the chemistry itself. So to solve that problem, what we did, we developed clickable fullerene building blocks. So they are very easy to prepare. You prepare a malonate here with 2 azide groups. The malonate is very simple, and you can produce the fullerene hexaadduct in very good yield, as you can see. And you control perfectly the position of the 6 atoms. The next step is the post-functionalization, and we use the so-called click chemistry, the copper-catalyzed alkene azide click chemistry. It's very efficient. It's working really well. You have to be very careful and use very pure material because you are grafting 12 times a functional group onto the same scaffold, so it's very sensitive to the purity, but the yields are pretty good, and we can really easily control the completion of the reaction. You can also reverse the reaction. You can produce an hexaadduct with 12 TMS protected alkynes. You can remove the protecting group and perform the click chemistry, but it's better to remove the TMS group in situ by just adding T-BAF to the click mixture, and you can graft whatever you want after. You are not limited by the size. You see here on that slide, the example are just small dendrons, and the yields are really good. If you would like to prepare those compounds using classical fullerene chemistry, I think for the first generation it would still work, but for the 2 next one, the yield would be almost 0. So that shows that our chemistry is really versatile, and we can now produce whatever we want. Of course, you can also combine the two functional groups onto the same scaffold, and in that way prepare multifunctional materials. First, you click an alkyne onto the azide group. The TMS protecting group will survive under those conditions, and then you apply a second click reaction by just adding TBAF and an azide to graft the second group. So we use that chemistry for many applications, and I will just show you a few examples to show you how specific those hexaducts are, especially when compared to classical fullerenes. The first example is a system with 12 porphyrines. Or typically when you combine C60 with a zinc porphyrin, what you expect is photo-induced electron transfer upon light irradiation. In that case, if you look at the emission spectra, it seems that there is a small quenching, but it's actually not a quenching. It's just that lower emission intensity is due to excitonic interactions between the porphyrins. So to finally observe electron transfer, we got inspired by work of François Dietrich, who could show that we can prepare supramolecular fullerene porphyrin ensembles by using apical coordination of zinc porphyrins. So we prepare the fullerene derivative with an imidazole group to coordinate the zinc porphyrin of the fullerene hexaduct. In one case, the fullerene is an electroactive subunit. It will be an electron acceptor. In the other case, the fullerene will be just a scaffold. It's a completely innocent group. It's just here to help the 12 porphyrins together. Nicolas Arnold did some photophysical properties. So upon excitation of the zinc porphyrin, you have photo-induced electron transfer, as expected. It's not necessarily the porphyrin that is coordinated that is excited. You have a kind of antenna effect. Upon electron transfer, you have hole hopping. The positive charge is getting far away from the negative charge, and in this way you have a long-lived charge-separated state. And if you look in details, we have a system that mimics all the fundamental steps of the photosynthetic system: the antenna effect, the electron transfer, and a long distance between the negative and the positive charge upon electron transfer. We also use that scaffold to have compact electroactive system. C60 is a good electron acceptor, but once you graft 6 atoms on the fullerene, it's not an electron acceptor at all. Actually, the fullerene hexaduct is electrochemically silent over a large potential window. So you can simply use it as a scaffold. And that's the work we have done in collaboration with Fraser Stoddart, because the PhD student who developed all the chemistry on the hexaduct, Julien Hill, went in his group for a postdoc. So we prepared a system with 12 biologene units. And if you reduce the biologene and you follow that by spectroelectrochemistry, you can see typical signature of dimers. What's going on here? It's because the biologene in a confined space, they form dimers. So you completely change the shape of your molecule, pointing in 12 directions, it points in 6. So you reshape the structure of your compound, and that's perfectly reversible because it's electrochemically triggered. We also used a lot of our fullerene hexaduct scaffolds for the preparation of bioactive molecules. That's just one example of dendrimer, where we grafted small dendrons with ammonium groups at the end. They interact very nicely with DNA, And with my colleague Jean-Serge Rémy and Ilkirch, we could show that we can use them for gene delivery. We also prepared some glycoconjugates, what we call Fullerene sugar balls, and those compounds have been used for many, many applications, and I will show you a few examples. In nature, sugar-protein interactions are very important, and as you know, nature is very, very selective. And to be selective, nature is not simply making the difference between galactose, mannose, glucose, and so on, but very often the proteins interact with several sugars at the same time. In that way, the nature of the sugar, but also the relative orientation of the sugar is important. So if you want to compete with natural ligand of proteins, you have also to present sugars in a multivalent way and to observe multivalent effects. So first, we had to see if our chemistry is working, and it worked perfectly well. If you start from the system, the C60 building block with 12 alkenes, you can graft all kinds of sugar, azides in very good yield, even some fancy ones like amino sugars. It's really working perfectly well. Also, the sugar chemistry was done in collaboration with Sébastien Vidal in Lyon, now in Paris, with Stéphane Vincent in Namur and Philippe Compin in Strasbourg. You can also reverse the chemistry. You can start from the Fullerene azide, and you can graft the sugar alkene very efficiently. In that case, the chemistry is a little bit more difficult because very often we have precipitation of the partially clicked compounds. I think the problem is the solubility difference between the two starting material, and in the middle of the reaction you have to change the solvent, and very often we use microwave to complete the reactions. But when we optimize all those conditions, it worked quite well. The yields are not always terrific because the purification is not always very easy. So now, going to the biological properties, and that has been done in collaboration with Anna Berti in Grenoble. She has two lectins of the same bacteria, Pseudomonas aeruginosa. Just Google that name, you will see that it's a terrible bacteria that is responsible for many nosocomial infections. And we wanted to see if we can have good interaction with our Fullerene sugar balls and those lectins. The first one we targeted is the galactolectin, leg A. And as you can see, if you compare a model monomer and the glycocluster with 12 sugar, you see that you increase amazingly the affinity. And that's really a nice example of glycoside cluster effect. You can clearly understand that why, because if you look at the protein, it's a tetramer and you have on each side two binding pockets, so you can imagine that you have a chelate cooperativity. Unfortunately, we had precipitation and we couldn't perform some ITC measurements with that system to have thermodynamic data. But it was possible with the second target, which is the fucolectin of the same bacteria, leg B. And in that case, we could perform ITC measurements. And you see that you have a weak multivalent effect. Before it was huge, here it's very small. So why is it like that? But if you look at the protein, you see that you have 4 binding pockets, but they are very far away. On each corner of that square, they are located, and there's no way to have a chelation. Actually, you have some aggregation, and the weak affinity enhancement is due to a high entropic penalty because of the aggregation. But of course, within the aggregates, you have chelate cooperativity as well. But Anne is in her group, she had another lectin from another bacteria that is as sympathetic as the first one. And in that case, the lectin is a trimer, and you have 6 binding pockets. But this time, they are all located on the same side of the protein. So if you look at the binding, now we have better enhancement. It's by 2 orders of magnitude. Before it was just 1. But already the KD of the monomer is quite high in that particular case. But it's clear that most probably we have some chelation, and that's why we have that enhancement. But 2 orders of magnitude, that's not much. So why is it like that? Of course, if you look the entropic part, it's fine. But if you look at the enthalpic and the entropic part, you have a penalty because of the clustering. And when you have bigger system, you have more clustering, so the entropic penalty is much higher. So that's always a limit. And of course, at the end, both are contributing, enthalpy, entropy, and if you have an entropic penalty, you pay the price for it. So is it worth going to big structure? We see that we have a limit. At one point, we have entropic penalty effects. But you know, we are chemists and we want to see how far we can go. So Philippe Compin prepared some dendrons with 3 immunosugars, and we could graph them. So we have 36 sugars. We went even further to the next generation, starting from a dendron with 9 immunosugar. We could obtain a system with 108 peripheral immunosugars. And in that particular case, we could find still some positive effects on the biological properties. But I will not enter into details. But if you look at those systems, you can imagine that it's a huge effort to produce a few milligrams of those compounds. There are too many steps, and it's quickly a big limitation for the chemist. So we asked the question, how to increase the number of sugars, but without being limited by the synthesis. Actually, we have all what we need. We just used Fullerene hexaadec building blocks. So we developed a macronomer with 10 peripheral sugars, an azic group, and you simply click it on our Fullerene scaffold. And at the end of the day, you have a first generation dendrimer, but with already 120 peripheral sugars. That compound was tested for its antiviral activity. That's a collaboration with Nazario Martin, Raviroro, and Delgado in Spain. And just to show you how that test is working, I prepared a small movie. So the Ebola virus is using a protein to enter in the cell. It's a modified Ebola virus that contains the luciferase gene. So if the cell is infected, it will become luminescent. Our compound has a very high affinity for the receptor and prevents the virus to enter. And you see the IC50 is very low, it's 0.4 nanomolar. So was it worth to use 120 peripheral sugars? The answer is yes, because if you just use a simple Fullerene model compound with 12 sugar, it's 1,000 times less efficient. And if you just use 1 sugar, it's 1,000,000 times less efficient. And it's really a molecular effect, because if you use a glycocluster, when you replace the mannose groups with glucose, you have no activity at all. So it's really because our compound has an ultra-high affinity for the DC sign receptor that the Ebola virus is using to enter the cells in that particular in vitro test. So using the same chemistry with Philippe Compin, we prepared a system where you have 120 peripheral immunosugars. So the chemistry is quite simple, it's very quick, so you can use very fancy peripheral groups. It's not a simple sugar to prepare an immunosugar like that. You have already 10 synthetic steps, so you have to be short and efficient if you want to go to very big structures like that. And the biological activity of those compounds were studied by Carmen Ortiz-Mele and her husband in Sevilla in Spain. So they tested it against several glycosidase, and the best results were obtained with the JAK-BIN alpha manosidase, which is very sensitive to multivalent effects. And if you compare our big guy with smaller model compounds with 12 sugars only, or immunosugars, or with just a monomer, you see that you have a huge enhancement in the inhibition. That means on a per sugar basis that one immunosugar in the mega molecule is almost 900 times more efficient than the monomer, and in the 12-mer, it's 150 times more efficient than the monomer. So even going to very large molecules like that, you can still have positive effects in the biological activity. So you have seen that we went from 12 sugars to 120. We developed the chemistry for it. It's very efficient. Our Fullerene compounds find many applications. So we have seen in biology, in particular with the glycoclusters, but also in material science. And what I would tell here is that we could perform all those applications because we first developed the building blocks and the chemistry. And that's the main message of the talk is something that François Dietrich told me when I was student or when I was postdoc in his group is, if you want to do something, first develop chemistry, after you can do whatever. But to finish, I would like to thank all my collaborators. I think I mentioned them during the talk. We have to thank all my students. And in the French system, that is not perfect, but we have the big advantage. We have permanent people, and I'm lucky and fortunate to have 3 outstanding permanent people in my group, Uwe Hahn, Michel Oller, and Yvonne Nierengarten. I have to thank also all the funding agency for the money, because that's important for our research. So if you want to follow the news of our group, you can follow us on Twitter. If you like small scientific movie, you can also go on our YouTube channel to watch them. And thank you very much for your attention.
Thank you very much, Jean-Francois. Let me start with sharing my screen. If you can unshare yours,
Yeah.
I have to. Zut. Can you unshare yours, and I have to give you something?
Yeah. Okay, now it will be
now.
Yes.
Yes. Yes. Yeah. Okay. So I wanted to remind everyone that Jean-Francois receives a prize today. He receives the DCO prize, which is the most prestigious prize of the division of organic chemistry. So for that, you receive this medal. So you see it on the screen, and it is here in my. What you see? I don't know if you see. petit galage. And here, up. Yes. So this is your medal, Jean-Francois.
Thank you.
So we will post it to you. Congratulations.
Thank you very much.
So there are, there are quite a few questions that are appearing on the chat, but I will try to have families of questions. Questions about the chemistry, and maybe questions about multivalency. So first questions about the chemistry. As you said, it's the root of your work. So the questions I would say turn around: is it possible to add different functions? So you showed at the end that you could add one different function out of the 6. But can you, for example, add 3 plus 3? Can you add 1? And is there an order in the reaction? Can you have the first grafting, then where happens the second one, and can you control first, then second, then maybe third, and etc., etc. So.
Yes, it's a good question. Of course, to have all the 6 atoms identical is easy. It's a one-step process. What is very easy to prepare as well is the 5-1. You just prepare a monoadduct that we know how to handle that, and then you complete with the 5 others. If you want to have the 4-2 and the 3-3, then it's more difficult. We did it. We developed some chemistry to have the 2 or 3 first ones at the right position. That's the big problem. That's the big issue. Because if you have, when you have one substituent, there's no stereochemistry issue. When you add a second one, you have 9 different positions, but only 2 are of interest to prepare the hexaadduct. So you have to be you have to develop first chemistry to prepare the right bis adduct to go to the hexa. It's the same if you want to have a tris adduct. We also developed nice chemistry. We use a tripod and we perform microcyclization on the fullerene. The first reaction is intermolecular, the second one is intramolecular, the third one as well. It's the only way to control the stereochemistry. I've seen that some people are asking about chirality. The hexaadduct, when all the atoms are identical, is not chiral at all. Of course, when you put sugar, it's chiral, but it's not an inherent chirality because of the fullerene. But there are very nice chirality problems in fullerene chemistry, and François Diedrich was one of the precursor in that field. If you just put 2 atoms on the fullerene, there are some bis adduct with a C2 symmetry. When you have 3, you can have C3 symmetrical tris adducts, and they are of course chiral. And we developed some chemistry to produce those tris adducts in an optically pure manner by using chiral tripods, optically pure tripods.
In terms of mechanism, because you seem to say that if you put a first cyclopropane, if you put a second one, you can have different regioisomers that will lead or not lead to the hexa functionalization. So that would mean that the hexa functionalization happens in some kind of equilibrium. The reaction is reversible apparently, and it leads to the most stable compound. Or
you have to work. Yeah, it's a good point. You have to work under conditions under thermodynamic control. When we prepare regular fullerene monoadducts or bis adducts using the cyclization trick, we don't work under thermodynamic control, but under kinetic control. Okay. You have to find a trick to have a reversible reaction. And among all the possible hexaadducts, there are plenty of possible hexaadducts. The symmetrical one we obtain is the most stable because you isolate 8 benzene rings, so you have 8 times the local aromaticity. And if you can introduce an equilibrium, then you can control the position of the 6 arenes. The first trick is you use, that's the trick of Andreas Hirsch from Erlangen, you use dimethyl anthracene. So you have a Diels-Alder, retro-Diels-Alder. You put a large excess, so you occupy the 6th function at the end, and then you replace them progressively with malonates. The other trick is to use a huge amount of CBr4. You generate bromoform in the reaction. The addition of bromoform to the fullerene is reversible, and that's the way you can have that equilibrium.
Okay, thanks.
It's under thermodynamic control. It's dynamic covalent bond formation.
So another series of questions deal with more the biology. So I will try to give you all the questions in one shot. Maybe there are not all of them, and then you can look at the chat. But so there are questions about the solubility of your, I guess, the glycodendrimers and stuff like that, and the question about metabolization of the C60 in the body. Maybe you answered this question, but I know for other scaffolds who bear sugars, the proportionality of between valence and activity, so inhibition, for example, is not always the more you have, the best it is. I mean, in your case, it seems to be the case, but in other cases, it's not. And finally, did you try to replace C60 by carbon nanotubes to get even more valency? So I leave you with all those questions. I'm sorry.
Yeah, of course, the bigger is better is not always the case, that's for sure. It's in some cases you are limited by negative anthropic effects, for sure. So it's not always necessary to go to very big structures. One question we have very often is about the toxicity of those compounds. I can tell you that there's all the assays we have done have shown that there's no toxicity. But biologists don't like too much fullerene. So they don't like nanotubes as well, because that's even more problematic because you don't control the structure. The advantage of our big molecules, it's a single molecule. You know exactly what you use. So to answer that question, we replace the fullerene core with a more simple core, that is the pillar 5 arene. It's a microcycle where we can graft up to 10 functions. But I didn't talk about that today.
Okay, thanks very much, Jean-Francois. I think if you can, another question. Why fullerene functionalization reduced its electron accepting ability?
You kill a double bond in the fullerene scaffold, and each time you kill one, you push a little. The reduction is a little bit more difficult. And if you look at the hexaelect, when compared to fullerene, you have 1 volt of difference for the first reduction.
Okay. So thank you very much, Jean-Francois. We will stop. If you look at the chat, there will be other questions, but now don't look at it yet, because if Thomas can come back and we will... so first, thank you. And so we'd like to have a little chat now with both Thomas and you. If Thomas, you can come back online.
I am.
Yes, here. Thanks very much. So it's an experiment that we are trying to do, a discussion with a little bit more personal advice or input from both of you. Don't go away, Thomas. You're frightened? So, okay.
No,
actually,
no, I just wanted to close the door.
We didn't do it on purpose, but I think you're a perfect example for our first question. I mean, Thomas and Jean-Francois, you both worked with François Diedrich, the same scientist, and now one of you is doing C60 chemistry, let's say, and the other one is doing DNA chemistry and epigenetics. So completely different fields, let's say. And so, so the first question, maybe to start this discussion, is how do you choose a subject in a career? How did you choose yours first, and what advice could you give to younger academics for a subject? How to choose a subject or a subject of investigation?
I can start.
Answering?
Yes, go ahead.
When I obtained my CNRS position, I had a good advice of François Diedrich. He told me, look in your working place what people are doing and just avoid to do the same. Try to bring something different. And for sure, when I arrived in France, there was no Fullerene chemistry at all. And we also worked a lot in the beginning on dendrimers. There were some excellent groups in dendrimers, but they were all in the south part of France, in Bordeaux, in Toulouse, particularly. But there were two dendrimer chemists in Strasbourg. So I think, you have to do something different than your colleagues, and you have to develop your own building blocks.
Yeah. So, but why did you choose? It's because you carried on what happened in your postdoc with François, basically.
Yeah, but in the group of François, I was mainly working on new ways to functionalize Fullerene. When I arrived in Strasbourg, I used Fullerene chemistry to produce materials. That was very different because I obtained my initial position in a materials science institute. And my deal with my institute was, okay, we cannot tell you what you can do. You are the chemist, but try just to interact with people.
Okay. So what about you, Thomas?
Oh, that's hard. The rate limiting step in science is very often creativity. And already Einstein said that imagination is more important than knowledge. So it's very, very difficult to be creative, to teach people to... I mean, I work on things that I neither did during PhD, nor postdoc, nor with François. So I was just interested in DNA chemistry. And then I think the only advice that I can give is people need to read. I mean, they need to read Angewandte, they need to read science, they need to read Nature, they have to read chemical and engineering news. So read, and then you find what is a hot topic, and then I would simply say, whatever you need to become creative, just do it. Think about the project and try to get an idea. I mean, at the end, the idea differentiates you from all the others. If the idea is good, then in the sea of scientists, you will be seen. So that would be my advice. And we have now started a Corona project in a very similar way. And we have wonderful data. We can kill 99.97% of the virus. Yeah, this is what we need, right? We scientists have to solve the problem. So it's no question that it's a hot topic. It's no question that chemistry is needed. So this is a good example. Just go into the literature, see what has been done, and try to come up with a good idea. And here you are. I mean, that's how I always worked.
How did you go to DNA concretely? You came back from Diedrich. Well, you got a position, and what you just stopped. You went 6 months in a library. You read everything that existed, and then you decided to go in this, or?
No, my way was different. I mean, I did my PhD with Stab at the Max Planck Institute, working on porphyrines and photosynthesis model system. At that time, this was super hot. Deisenhofer, Huber, and Michael, they got a Nobel Prize for solving the structure of the photosystem. So that was what. And then I went to Riebeck for a postdoc at MIT, and we worked on self-replicating systems, DNA, non-DNA self-replicating system. And here I got in touch with the idea of mutagenesis, of faithful translation, copying of genetic information. And then I read a lot about DNA repair, and for which actually a chemistry Nobel Prize was given a few years ago to the discoverers of DNA repair systems. And then I said, okay, what can I do as a chemist? Well, as a chemist, I can synthesize DNA lesions. Sounds a little bit odd. Then I can incorporate them into small oligonucleotides, and then I can study what are the physical effects of having a lesion in DNA. How does it destabilize the duplex? What are repair enzymes doing with it? And blah, 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 blah. And hydroxymethylcytosine was at that time recognized as a DNA lesion. So we basically were dragged into the field when all of a sudden it was discovered that hydroxymethylcytosine is not a lesion. But so everything comes from DNA repair. And with François, I had at that time an assistant professor position at ETH. And as an assistant professor, you were always under the umbrella of a big guy. And that was him. So he was watching that this little chicken, Thomas Carell, is developing well, and but he did not interfere with my science.
Okay. Thanks. So we have some questions on the chat. There are final PhDs that are worried, and they would like to know, what would you look for in a candidate if you were hiring a postdoc in the near future? I think this is an important question. And will you actually hire postdocs considering the situation? That's another question.
So maybe I'm asking, we are always looking for good co-workers. I mean, typically, I have a lot of applications. I cannot take everybody, but I can promise that I look at every serious application and look carefully. And typically, I make a money offer. So I tell the person, listen, if you come, I will secure funding that you get. But of course, I'm asking that we both together try to get a fellowship. So I'm looking always at the scientific pedigree. Where did the person study? Is that a good university? Where did he do a PhD? Is this a good and solid colleague of mine? And third, are there publications? So has the person proven that he can do science and publications? And then typically I take the person, and then we apply for a Humboldt fellowship. And in most of the cases, this Humboldt fellowship was awarded, and then the person can start in my group to work as a postdoc. Typically, the groups stay in my group a little bit longer because the techniques need some time. Needs some time to get acquainted with these techniques in water, and then they go either into industry. Some people are with BASF and Bayer, or they start an independent group, and then they are allowed to take the research that they did in my group with them to basically develop it into a new direction. This is what François was teaching me. So I think it's relatively easy, and I just accepted a new person from Spain. He indeed got the Humboldt Fellowship, and today, after this conference, I will get in contact with somebody from Britain who has applied to my group, and I will make him also an offer. So I like to get applications, and then I can look at them, and the more applications I get, the better it is.
Okay, there are 400 people looking at you, so what about you, Jean?
Yeah, okay. I think there is not much to add to what Thomas told. It's true that, of course, you look where the people prepared the PhD, what they have done. What is important is typically when you have an interview with people is you have to feel the enthusiasm for science. And that's something essential. You need to have coworkers that are fascinated by what they are doing. I think science is passion.
Yes, absolutely. It's not money anyway. So,
yeah. Of course, it's money. Money is super important. What would we do without it? Yeah, but this.
Okay.
This is our job, right? To organize money.
So just maybe a last question before we switch to the other session of talks. What is your publishing strategy and communication strategy in general? What would be your advice for younger researchers?
Jean-Francois, do you want to start now?
Oh, I can. Okay, for me, my strategy is simple. I like to have solid papers. I want to have everything well done. It's a pleasure. It's not a rush. It's not a race. Not to publish whatever. I hate papers where you have just a few data and that are not well done. It's important. The quality is important. Then the choice of the journal, of course, everybody would like to publish in the best journals. I'm referring a lot of papers, and I can tell what I'm putting in my reports is something I'm applying to my papers when I select the journal. I have a quite transparent publishing policy. I'm in Europe. I'm a part of the European Chemical Society. And I publish most of my papers now in the European journals, in Chemistry Europe journals. These are the journals of our society. And I think it's very important for people to publish there. And an advice to young people, don't publish in rubbish, predatory journal, or sometimes wait a little bit more to have a little bit more data to publish in a better journal. Don't be in a hurry. Think in terms of quality.
I fully agree with Jean-Francois. We should not publish the least publishable unit, but publish nice stories. And I like to publish. I mean, I must admit, I'm a big fan of having every 1, 2, 3 years a paper in Science and Nature. It's a lot of work. It's not that you publish there. It's really terrible. I mean, you fight with reviewers forever. But I like to do it because at the end of the day, you get so much visibility. And of course, the people who are first authors on their paper, on such a paper, their career is made. So I have always this fight between, let's publish this in Angewandte and JAX, which is super nice. And then I'm saying, well, if we do this and this and this, we can go a little bit higher. And then it's a little fight with the students. Some students want to publish very quickly because they want to go on, and others would like to publish higher. And so this is a lot of fun and we do it in a very nice way. But and then of course very often the paper is not accepted, and that's also clear. But yeah, I like to do this. I like to. I like to play the games. I like to play it, and And the last paper, 2019, had at the end an altmetric score of 350. It went into FAZ, Süddeutsche, El País, in all these journals. So I really like that. Yeah. It's a boost for the people on the paper. It's a boost. More for them than for me.
So aim bigger, basically. That's your advice. Okay, thanks you guys. Thank you for this nice chat. It's now time to go back to science with slides, etc. And thank you very much for this discussion.
Thank you, Matthieu.
Okay, so our next speaker is Xavier Bugaut, and so maybe I can present him with his. No, I will present him with his prize afterwards. So I give you.
Thank you very much.
Word.
Yeah, thank you very much, Matthieu, and also thanks to Thomas and Jean-Francois. I think that was very nice session and some insightful comments and recommendations from your side. I will now introduce Xavier Bugaut, who's the next speaker of this event. So Xavier studied chemical engineering in Montpellier and received his master in chemistry at the University of Montpellier in 2006. He then moved to the ICSN in Yves-sur-Yvette for his doctoral studies, and he received his PhD in 2009 under the supervision of Emmanuel Rounon. After a postdoc research stay with Frank Glorius in Münster, he took on a position as Maître de Conférence en Chimie Organique at Marseille University in 2011. His research focuses on enantioselective organocatalysis and the control of axial catalysis, and he also works on multi-component reaction and the synthesis of natural products. He is the recipient of the Journeé Prize for Young Lecturers, which was given out last year by the French Chemical Society, so by the DCO, the Organic Division. And in his award lecture today, he will tell us more about the asymmetric synthesis of atropisomers. So Xavier, we very much look forward to your talk, and I would like to ask you now to share your screen. Thank you. Perfect. Yeah, we can see your slide.
Okay, so thank you very much, Anne, for the very nice introduction. And of course, I'd like to say thank you to the Organic Chemistry Division of the French Chemical Society for the Jean-Normand Prize. It's a very big honor for me. And also, of course, the talk should have been in last April, but it was not really possible. And I'd like to thank EurJOC to also allow that we organize this online meeting instead. So to start the talk, I'd like to give you just a brief overview of what we've been doing in my group over the past few years before really focusing on the topic I want to discuss today. So basically, what we started to do was to do chemistry with original dicarbonyl substrate, and thinking that we could combine the functional group tolerance of enantioselective organocatalysis with the advantages of multi-component reactions. And with this, targeting important molecules like nitrogen-containing heterocycles, and always keeping an eye on the selectivity issues, either being regio, enantio, and diastereoselectivity. So that led us to the development of different complementary research directions. At first, synthesizing original beta-dicarbonyl substrates, then using those substrates, especially ketoamide substrates in enantioselective organocatalysis. For example, we could use these beta-ketoamides in micro additions where they could offer diaster control, which is not possible with ketoesters. And then going to more complex reactions, so multi-component reactions with those original substrates. More recently, we became interested into the synthesis of atropizomers. That would be the topic of today's talk. And also now with halogen bonding organocatalysis, which is still starting in our group. And actually, over the past few weeks, I've been mostly focused on another research topic, which is really different from chemistry, which is probably the most complicated, but also the most rewarding thing we could do. So, but let's go back now to axial chirality. And our idea at start was to look at these heterocycles, and we could prepare quite a lot of compounds with piperidine, piperazine, but not with pyridine. And so we became interested in the 4-aryl pyridine atropizomers because they could be found in quite a lot of compounds, either natural product or synthetic compounds, with very interesting bioactivities. And when we started to work in this field, there were no enantioselective synthesis for those compounds. They were either just synthesized in a racemic fashion or separated through Carell HPLC, but no enantioselective synthesis. And our idea was just to start from this textbook reaction, so the Hansch-pyridine synthesis, where the dihydropyridine could be assembled through a multi-component reactions, and then the pyridine was obtained by oxidation. So our simple idea was, is it possible to prepare enantioenriched dihydropyridine with central chirality, and then proceed with the oxidation of those compounds to the 4-aryl pyridine, by having a central-to-axial chirality conversion. So, of course, we did not start from scratch. There were already things present in the literature. Actually, the original hypothesis for this conversion of chirality comes from 1955, so it's quite old hypothesis, even though more than 30 years were needed to obtain the proof of concept in the work of Meyers, where he could carry out a diastereoselective dearomatization of this quinoline motif, and then re-aromatize it through oxidation. And we could see that the good diastereoselectivity in the original Michael addition, in the original dearomatization, could be conserved through the enantiomeric excess of the final product. When we started, there was only one example of combining enantioselective catalysis with central-to-axial conversion of chirality in the work of Thomson, with an enantioselective rhodium-catalyzed Michael addition to a dienone, then dimerization and aromatization through elimination to get to the biaryl compound. So our first challenge was to find conditions for the synthesis of enantioenriched dihydropyridine that could still tolerate the steric hindrance required then for the stability of the final atropisomer. So this method developed by Singh using enones with the pyridine motif here was the best one, and using a bifunctional organocatalyst and then treating the Michael adduct with ammonium acetate, we could obtain the dihydropyridine with good yield and very nice enantiomeric excess. Then we proceeded to the next step, so the oxidation, and we soon realized that the conversion of chirality is strongly depending on the oxidant. Indeed, DDQ, which was the oxidant used by Meyers, gave us a very poor enantiomeric excess, only 8% enantiomeric excess for the final product. Quite a lot of other oxidants were inefficient, and the only good one was manganese dioxide, which could deliver the final product with 86% EE. And we could secure the absolute configuration of the final product through crystallization. And what was also nice that this crystallization could occur with an enantioenrichment so that we could obtain virtually enantiopure compounds. So I will not detail the scope of all the reactions I'm presenting so that I have more time to discuss about completely new results that were not published yet. So when developing this reaction, we also realized that the oxidation process was really sensitive to the solvent. And starting from this S-dihydropyridine, we obtain, as I showed before, the AS compound if we do the oxidation in cyclohexane. But then if we use an acidic solvent, we could go from the same enantiomer of the dihydropyridine to the other enantiomer of the final atropisomer. Just a few data here. So acidic acid delivered the product with only moderate enantiomeric excess. And if we go to trichloroacidic acid in combination with acetonitrile, we could obtain a little bit better. So it's still not very interesting from a preparative point of view, but for the understanding of the whole process, it's quite an interesting observation. Also, if we put a protic solvent, then we just kill this inversion of stereoselectivity during the oxidation. A last point on this is that this inversion is obtained only when we have Lewis-basic functionalities at the proximity of the stereogenic axis. So how does this all work? So at first we checked that the final atropisomer is configurationally stable in the reaction conditions. And if we look at the dihydropyridine, we can see easily that for one enantiomer of the dihydropyridine, we have two different conformers. So this can be seen in NMR. We have an exchange here at room temperature. We could slow down this exchange if we go to low temperature and characterize those two conformers that we could call exo or endo, depending on the position of the methoxy group. So those two are in fast equilibrium with a half-time life of less than 1 millisecond. And we can see also that the ratio is of 2.6 to 1. So it's not enough to explain the selectivity. That means that, in fact, we need to have a faster oxidation of one of the conformer to get to a major enantiomer of the product. What we still don't know is why manganese dioxide is able to provide a faster oxidation of one conformer over the other and not the other oxidant. Also, this picture might be a little bit more complicated. We carried out calculations so we could find the same theoretical equilibrium constant than the experimental one. But those DHPs, in fact, they exist as dimers in solution. So we could see it through calculations and also with the optical rotations, which becomes higher when we decrease the concentration of the sample. So that makes it even more difficult to understand what happens during those oxidation process. When we go to the acidic condition, this time we obtain the other enantiomer as the major one. If we look at the protonated species here at the pyridine, we have once again an equilibrium between those two species, but that goes more on the left side. So that does not seem to explain why we reverse the stereoselectivity. But there's something interesting also we've seen in the calculation is that now we could have a rotation around this carbon-carbon bond between the two rings, and we could access other conformers. So there's probably something going on between those functionalities with some kind of hydrogen bonding interaction we still need to figure out. Those conditions were quite interesting, but we needed to use more than 20 equivalent of manganese dioxide. So we wanted to go to greener oxidation conditions. We realized that the dihydropyridine can oxidize readily with air, and thus this oxidation becomes faster if we put a base in the reaction conditions. By doing this, using a strong base such as sodium terbutoxide, we could obtain a rather correct yield of the product and a very encouraging enantiomeric excess. And through the optimization of the conditions, the key point was to use dioxane as the solvent. In this case, we obtain an enantiomeric excess that's even higher than we obtain with manganese dioxide, with conditions that are a lot easier to use. We carried out some mechanistic studies on this. At first, we can say that the deprotonation occurs at the NH. We could see this through NMR. And then EPR studies with spin trapping showed us that we have a carbon-centered radical intermediate in this process. To finish with, I will present an application of this strategy to a useful molecule, so this DPP4 inhibitor, for which it was shown that the two enantiomers have quite a different biological activity, so it could be interesting to provide an enantioselective synthesis. So the retrosynthesis would go through some functional group transformation, and we could apply our strategy of central-to-axial conversion of chirality, starting from this dihydropyridine that could come through an enantioselective Michael addition. In the synthetic way, so we can see that we can carry out the Michael addition, which was unprecedented, using this chiral chloramide catalyst. We obtain the Michael adduct and treated with ammonium acetate in a refluxing azelaic acid. We obtain the corresponding dihydropyridine with the very good enantiomeric excess. For the oxidation, we started, of course, with the conditions that we had before. So manganese dioxide was unfortunately really bad in terms of enantiomeric excess, and the basic conditions in air provided no reaction. So we had to screen again other oxidants, and the best one was this oxoammonium salt, which could give the final product with an enantiomeric excess that's still not perfect, but it was enough to complete the synthesis. The functional group transformations go through the chemoselective reduction of the ester to the alcohol. Then we can transform it to the azide using DPPA in basic conditions, and Staudinger reduction to the final product with only a minor erosion of the enantiomeric excess. We could also do other examples by replacing this aromatic group by other ones. And we soon realized that there was a tendency for an increase of the enantiomeric excess of the final product with the size of the substituent. To see this better, we normalize the result by introducing a conversion percentage, which is the EE of the final product divided by the EE of the starting material. And like this, we could find a linear free energy relationship between a Charton steric parameter and the logarithm of the conversion percentage, showing that it's really the size of this substituent that will discriminate for the enantioselectivity of the final process. So I'm done with the science I wanted to show you. So I'd like to thank all the people who participated in this work and all the funding organizations. So all the people highlighted in red are the ones that were involved in what I presented today. And to finish with, of course, all the students that were involved in this project, especially Ophélie, who started all this, then Tanguy, who took over, and Kartik, and the current student working with me. So today I presented notably the work of Arun and Clément, and the two other students, David and Bogdan, they're working on the other topics, so the halogen bonding organocatalysis. So I'd like to thank you for your attention. And of course, I'd be really happy to answer your questions.
Thank you. Thank you very much. Can you...
yeah, I can stop sharing the screen. Okay.
So I just want to give you virtually your medal for the Prix Jean Normand. So it is in my hand.
Yes.
I don't know if you can see it, not yet. It's here, so we will send it to you. And congratulations for this prize. Yeah.
Thank you very much, Matthieu.
So for the sake of time, I will ask you just one question around the kinetics of your reaction, which seems to be really important. So, well, apparently not, probably because you would talk about this, but is electrochemical oxidation would be an interesting thing to try to study the kinetics of this reaction and to understand more the mechanisms.
So actually, I've started to discuss with Claire Favre in Paris about electrosynthesis and its use for this kind of oxidation. So normally in the coming weeks, we should try to start doing things about this.
Okay, so just another question. Did you think about using racemic dihydropyridines and oxidizing them with Carell oxidants?
We're doing this at the moment, so that's still don't have any result to present, but of course that would be an even more interesting strategy to do this, because it's a lot easier to prepare a racemic dihydropyridine than enantioenriched dihydropyridines. So it's really, the reactions are running at the moment, so Clément is working on this.
Okay, so let's thank you again. There are some questions on the chat. Please look at them, and thank you very much for your talk, and congratulations again for your prize. So yeah. So next.
Okay, so now we will continue with our next speaker, and that's Ludwig-Maximilians-Universität. So Ludwig, if you can come on screen and turn on your camera.
I think it's fine. Hi.
Thank you very much. Welcome. So Ludwig studied chemistry at the INSA in Rouen, and he received his master in chemical engineering in 2011. He then pursued a PhD in organic chemistry under the supervision of Fabrice Ondobel at Nantes University, which he completed in 2014. After a postdoctoral stay with Harry Anderson in Oxford, he returned to France, where he became CNRS researcher in Rennes in 2015. So his research interests lie in the study of chiral pi conjugated molecules for optoelectronic applications. He received the Marc Julia Award from the French Organic Chemistry Division last year, and he will present his award lecture today entitled A Polarized Spotlight on Chiral Molecular Materials. So, Ludwig, please share your screen, and we look forward to your lecture. Thank you.
Yep. So here,
yeah,
everything is fine.
Yeah, just put it in the presentation mode.
Yeah. In big screen. Perfect.
1 second. Yes, now you can go ahead. Thank you.
Okay, thank you, Anne, for this very kind introduction. And I would also want to thank the DCO and the EurJOC for setting up this web seminar, and of course for the prize. It's, as for Xavier, it's a real honor and strong support from the community on the research we have been doing in Rennes in the properties of chirality, as Xavier again, but more focus on the potential of chirality in optoelectronic applications. So indeed, most of you know that a chiral molecule interacts specifically with polarized light, which results in different response for each enantiomer of the chiral compound, which are of strong importance, for instance, to characterize the enantiomer purity of a drug or of a reaction, or the conformation of a protein. So usually, this kind of characterization is performed by recording the electronic circular dichroism of an enantiomer-enriched compound, which correspond to the difference in absorption between a left and a right circular repolarized light for a given enantiomer, with the opposite response for the other. So much less popular, the equivalent process in emission, namely the circular polarized luminescence, represent the emission of either a left or a right circular repolarized light from an enantiomer upon excitation by an unpolarized light. Finally, the intensity of these two polarization process can be evaluated by the dissymmetry factor J, so either G-abs or G-helium, which is equal to -2 or +2 for full right and left polarization, respectively. So recently, the possibility to generate circular repolarized light by a chiral compound has received renewed attention to the potential of a CP light in several domains of application, such as in display technology, in spintronic, or in chiral bioimaging. As organic chemists, our objective is to design chiral molecule possessing intense chiroptical properties to further explore the potential of CP light in practical application. Theoretically, the intensity of the dissymmetry factor can be related to the intensity of the electric and magnetic dipole moment of the emission transition and the angle between these two vectors. So from this relation, it's easy to see that high value of CPL will be reached for molecules showing large magnetic transition dipole moment, as in chiral lanthanide complexes, or in liquid crystals compounds owing to their chiral supramolecular arrangement. However, these compounds often present low luminescence quantum yield and poorly tunable optoelectronic properties. For these reasons, organic molecules have appeared as an interesting alternative in addition to the easier integration in practical devices. However, they usually exhibit very low CPL intensity, with helium ranging from 10 to the -3 to 10 to the -4, with few exceptions based on helical shape derivative, displaying helium up to 3 or 4 10 to the -2. Accordingly, understanding the molecular factors that govern the CPL efficiency is currently needed to enable further development in this field and increase the CPL intensity of small organic chiral molecules. So our group in Rennes contributes to this topic by developing innovative chiral compounds based on carbo-6 helicene, which present an intriguing helical p-conjugated system and already intense chiroptical properties such as high optical rotation, high electronic circular dichroism signature, and already CPL response in the classical range of organic chiral emitter. So by using either organometallic approach or strategy based on the functionalization of helicene bis ethinyl derivatives with organic achiral dyes, we try to expand these properties toward the visible and near infrared region of the spectrum and explore chiral optoelectronic applications. In fact, during the next part of this presentation, I'm going to show you our obtained results following the later strategy, and notably its potential for chiral OLED application. So the work I will present has been mainly developed by Kees Dababy, which was a former PhD student in our group. As a first example, he decided to prepare pi-conjugated chiral diketopyrrole helicene dyes of different size and investigated their photophysical and chiroptical properties. So the DPP dyes was selected for its intense and red luminescence, as you can see here, which give the possibility to design innovative red CPL emitters. So the UVVs spectra of these compounds show strong absorption in the UVVs region and between 500 and 650 nanometers that correspond to the helicene and the DPP units, with similar signature for the three compounds and intensity proportional to the number of the DPP units in the molecules. Interestingly, all these transitions are active in circular dichroism, and even the ones centered on the achiral DPP units around 600 nanometers, which clearly reflects an efficient synergy of the DPP-based transition with the chirality of the helicene backbone in the ground state. Moreover, what we can see here in this region is an unexpected dependence of the intensity of the ECD signals on the number of substituents of the helicene unit. As you see here, when the helicene is symmetrically functionalized by a DPP unit, here in this H6-DPP2 or the oligomers, the signal is much more intense than for monofunctionalized helicene derivatives. So the structured luminescence of these chiral dyes, also rising for the DPP units, shows some circular polarization here. The obtained mirror image spectra for these three dyes resulted in gallium value up to 10 to the minus 3, with fluorescence quantum yield of around 40%. While this degree of polarization is in the range of classical organic dyes, the obtained CPL signatures are among the most redshifted reported to date for small chiral organic molecules. Interestingly, as in the case of the electronic circular dichroism, higher CPL values are obtained for symmetrically substituted helicene derivatives. Indeed, the DPP-centered pi orbital in H6-DPP2 are weakly interacting with each other across the helix, and we can consider them as two electronic dipoles within a chiral environment. And due to this chiral helicene, these two dipoles couple with each other and create a strong magnetic transition dipole that contribute here to the enhancement of the CPL intensity through a process called chiral excitonic coupling. Overall, these results show that a strong synergy between the chiral helical p-conjugated system core and the photophysical properties of the dyes can be obtained, especially when the helicene backbone is symmetrically functionalized. So, with this in mind, we decided to further explore this innovative strategy by replacing the DPP unit with naphthalimide fragments in collaboration with colleagues from Angers. Our idea was to use the chiral helicene as an electron donor and involve it more strongly in the photophysical properties, resulting in chiral push-pull dies with a dipolar structure for the monofunctionalized one and a pseudo-quadrupolar structure for the bis-functionalized one. In fact, this design gives rise to an intense intramolecular charge transfer bond arising from the helicene unit to the naphthalimide fragments, as observed in the UVV spectra of this new compound at around 410 nanometers, and in comparison with the isolated precursor. So this intramolecular charge transfer bond was also confirmed by theoretical calculation, and the HOMO, where the HOMO is located on the helicene backbone, and the LUMO on the two naphthalimide fragments. So the presence of such type of transition allows us to observe a positive solvatochromism in fluorescence here with a typical and similar for both compounds redshift of the emission spectra with an increase of the solvent polarity. Interestingly, such solvatochromism impacts differently the CPL response of 1 and 2. Indeed, the monofunctionalized helicene by one naphthalimide units show a constant CPL intensity in the 3 solvent with a Gaussian value around 2 to 3 10 to the minus 3. In the contrary, the P2 show an expected threefold decrease of the CPL intensity when going from non-polar solvent to polar ones. In fact, the intense structured CPL spectrum here in black in cyclohexane becomes broader, unstructured and weaker in DCM here in blue, and even weaker in DMF here in red. So thanks to a combined experimental and theoretical study, we rationalize this effect in terms of symmetry breaking of the excited state before the emission process. In fact, in apolar solvent, so the emission of 2 come from a almost equally distributed electronic density between each arm between each naphthalimine helicene arms, which favor an intense exciton coupling process, as in the case of the helicene DPP system, and ultimately lead to a high CPL intensity, for here the Gaussian of around 10 to minus 2. So conversely, the more intense reaction field imposed by polar solvents, such as DCM and DMF, induce a more localization of the excited state, preferentially on only one naphthalimide helicene branch, which, as a consequence, strongly decrease the exciton coupling effect, as experimentally confirmed by the measurement of the CPL response of the monosubstituted helicene naphthalimide, which does not present an exciton coupling processes. So it is worth emphasizing that compounds 2 exhibit in cyclohexane a very high value of CPL in comparison with reported value for chiral organic molecule in solution, which again highlights the benefit of chiral excitonic coupling strategy to enhance the theoretical properties of the helicene-based dyes. Further calculations show that for this system, the angle here between the magnetic and electric dipole moment of the emission transition is around 50 degrees, which is in fact not optimal for reaching higher CPL intensity, keeping in mind that the Gaussian factor is proportional to cosinus theta between the electric and magnetic transition dipole moment. So to reach an optimal control of this angle and given the obtained results, the high CPL value for weak dipolar character transition in cyclohexane, we recently developed a new generation of helical CPL emitter with low electronic dipole substituent. To illustrate this new approach, I only detail here the CPL response of two compounds bearing either a pyridine substituent or a phenylcyano-1. So as you can see here, the intensity of CPL show maxima corresponding to the unpolarized luminescence with very high values of CPL intensity for both the p enantiomer of the cyano and pyridine derivatives with gallium around 3×10⁻², which is a threefold increase in comparison to the bis naphthalimide compounds. So the theoretical analysis of the CPL reveals, of course, large electric and magnetic dipole vector, but especially a very small angle between these two vectors of around 5 degrees, which clearly contributes to the increase of the CPL intensity. So importantly, the obtained CPL values of these compounds are among the highest value recorded at the molecular level for organic molecules, and notably in comparison to other helicene or helicenoid derivatives. So the obtained results for this helical molecular architecture prompt us to investigate their performance as a CPL Carell emitter in top emission OLED devices, which represent a highly relevant and emergent OLED architecture for micro display applications, such as in camera, near-eye display, or medical analysis. So due to the vapor deposition technique used to construct the devices, we turn our attention to this new compound, since it also shows intense CPL response, but more importantly, we thought that the presence of the silyl group may help the vaporization process. Indeed, deposition of both enantiomer occurs smoothly with no racemization during this thermal vaporization. So finally, proof of concept CPU OLEDs has then been obtained and their optoelectronic characteristic investigated. As you can see here, the luminance tension characteristic of this non-optimized device show a clear diode behavior with a very modest external quantum efficiency, which is very small compared to standard top emission devices. But in our case, we were really interested by the fact that if we can have a polarization of the generated electroluminescence. So as expected, a similar electroluminescence spectra were recorded for both enantiomer, showing a non-structured profile with a maximum of intensity at 500 nanometers. So the measurement of the difference of intensity between left and right circularly polarized electroluminescence reveal, however, opposite response for both enantiomer in line with the obtained CPL value in solution, suggesting that the circular polarization is effectively induced by the Carell dopant. Finally, the plot of the electroluminescence dissymmetry factors agreed well with the gallium response in solution, confirming the origin of the polarized electroluminescence with a G of electroluminescence value up to 8×10⁻³ . So of course, it's not the value we got in solution, and in fact, in an ideal device, it should be the case. We should obtain the same degree of polarization. However, in our device, we know that some depolarization often occur due to the light reflection and polarization inversion at the counter electron. So while this work deserves further device optimization, we think that these promising results clearly represent an interesting example for Carell top emission based OLEDs. So to conclude, I hope I show you some interesting aspects of the use of chirality in molecular materials. So as you saw, a synergy can be obtained between the chiroptical properties of the helicene backbone and the photophysical properties of organic acyral dyes, resulting in intense and modulable chiroctical properties across the visible spectrum and specifically the circularly polarized luminescence. So more than only recording CPL signals, we also highlight the potential of CPL spectroscopy to investigate the electronic features of luminescent state in chiral system, which provide a deeper understanding of the CPL process itself and may open new direction for designing more efficient CPL emitters. Finally, thanks to a collaborative ANR project, we are now developing circularly polarized OLED, which nicely close the loop between our fundamental structural relationships properties and the application in real device. So to finish my presentation, I would like to warmly thank Dr. Jean-Francois and Kais Dabibi, which were strongly involved in this work, and especially Jean for continuous support in this topic. I just take this opportunity here to congratulate her for being the recipient of the DCO 2020 prize. Hopefully you will attend to her presentation in real next year. I want to also thank all the members of the PMM group that are all there, the OMC team here in Rennes and the Institut of Chemistry in Rennes, and more importantly, all our collaborators who help us to separate our enantiomers, so especially Nicolas Ventuin in Marseille, and investigate the possible application in device of the chirality properties or in CP-OLED or for the theoretical study. Of course, all the funding agency, again, the DCO for the prize and for this web seminar, and you for your very kind attention.
Thank you. Thank you very much, Ludovic. We have only time for one question that I will convey you right now. I'm sorry.
No problem.
So, what effect could be expected by increasing the size of the helicene? Would that be interesting? That would change the geometry as well?
Of course, it will impact the two vectors, the magnetic and electrical ones. There are a couple of studies on 8, 10, 14 helicene, even 16 helicenes that show that you increase the polarization, specifically in the electronic circular dichroism, but it's at the compromise of the synthesis, of course. So I would encourage people to work on the supramolecular chiral system, maybe to reach a very high degree of polarization.
So thank you for answering already this question, because that were the other question that you will see on the chat about supramolecular assemblies and maybe aggregation and stuff like that. So let me finish by presenting you your prize, the Marc Julia prize. So the medal is real. We will send it to you, and congratulations again, Ludovic.
Thank you very much.